Hi, my name is Jeff Hoffman, and in this lecture I'll cover the lab techniques that are useful to know for this step 1. Unless you're doing research or going into clinical pathology, you'll probably never need to know how to do any of these yourself, but it's still important to have at least a general understanding of a variety of lab techniques because you'll be interpreting and ordering these tests. Knowing more about them will help you order the right tests, be able to interpret the results, and most importantly, of course, do better in your board exams. So speaking of doing things better, I want to remind you that I want to make these lectures better every year. So please use the thumbs up and thumbs down buttons at the bottoms of your screen to give me feedback. Alright, let's talk about lab techniques. The polymerase chain reaction, or PCR, is used to amplify DNA. This just means making copies of DNA from what you already have, and you're only amplifying a specific piece of DNA that's maybe a few hundred base pairs long. So why would you want to do this? Well, first of all, if you want to see if a particular sequence of DNA is present in a sample, so for example if you suspect a patient has a viral infection, you could use PCR to try to amplify a DNA sequence that's specific to that virus, and if nothing is amplified, then that virus probably wasn't present in your sample. It's also useful if you want to take a closer look at the sequence of a particular site in the patient's genome. For example, if you want to look for a mutation or a version of a gene that might cause a disease. Okay, so how do we do PCR? Well, to start with, you need your DNA sample called the template, which is shown here, and you also need a heat-stable DNA polymerase, which is usually Taq polymerase, from the thermophilic bacterium Thermus aquaticus. You need nucleotide primers, which are complementary to the ends of your DNA template, and of course, the 4 types of nucleotide triphosphates, which are the building blocks for your new DNA strands. These are all combined together and put in a machine called a thermal cycler, which does the rest of the work for you. A thermal cycler, as you might guess from its name, cycles between high and low temperatures. The first step is to heat the DNA, which melts it, to allow the strands to separate from each other. Next, it cycles back to a lower temperature, which allows the primers to bind to the single strands of DNA. One primer is complementary to each strand of DNA. The only reason the primers bind to the template rather than the 2 template strands binding to each other is because the primers are much more abundant. The final step is DNA polymerase making a new strand of DNA, starting from the primer and using the original strand as a template. This results in 2 pieces of double-stranded DNA, which is twice as much as you had before, at least for that particular region. By repeating these steps a bunch of times, which is as easy as letting the temperature increase and decrease over and over again, you can end up with many orders of magnitude more copies of DNA than what you started with, because you double it every time. For example, 30 cycles would give you 1 billion times more DNA than what you started with, because 2 to the 30th is about 1 billion. Next, you'll want to make sure you amplify the right thing, which you can do if you know how long the DNA you're trying to amplify is. You can do this by running the PCR products through an agarose gel using electrophoresis, which will separate the fragments by size. Since you now have billions of copies of this particular small region of DNA, it's the only thing you're going to see. By comparing it to this DNA ladder, which contains fragments of known sizes, you can determine whether you amplify the right piece of DNA.